Okay, it's 10 o'clock, and I see people are coming in, so we'll get started. Welcome everyone. Thanks very much for joining today. This is the SBP Cancer Center seminar series, and today's topic is autophagy and cancer, and we've got two really great speakers lined up. I'm really excited to hear what they have to say. So my name is Peter Adams. I'm sure most people know me. And also want to introduce Swasti, who is the, although I'm hosting today's session, Swasti is the organizer and moderator of the series. And Swasti will be moderating the questions. So in terms of questions, I should say that if you could put your questions into the Q&A function of the webinar, not the chat. There will be time for questions after each speaker finishes, and then I think also at the end, and as I say, Swasti will moderate those questions. Okay, so our first speaker today is Christy Towers, from a neighbor from the Salk Institute just down the road. Christy has been an assistant professor at Salk since July of 2021. She was previously a postdoc with Andrew Thorburn at University of Colorado in Denver. And there she did some really cool work looking at mechanisms by which cancer cells circumvent autophagy inhibition. Okay, so obviously I think everybody knows that autophagy is a target for inhibition and killing of cancer cells, but like many therapeutic interventions, cancer cells have a nasty habit of getting around this and bypassing and acquiring resistance. So Christy defined at least two mechanisms by which cancer cells overcome autophagy inhibition, one through upregulation of NRF2 signaling, and another really novel, interesting mechanism, which I think Christy's going to talk about today. I'm really excited to hear more. And that's through a mechanism involving mitochondria-derived vesicles trafficking from the mitochondria to the lysosomes as a mechanism of mitochondrial homeostasis when autophagy and mitophagy are inhibited. So that's a really interesting novel mechanism. In her own lab, Christy is continuing to study adaptations to autophagy inhibition, applying optogenetic tools, and as I say, continuing to understand the role of these mitochondria-derived vesicles. In line with, so Christy is our rising star of today's program, and in line with this, Christy already has many awards on her CV, including a K99 R00, and was recently the runner-up in the New York Academy of Science Rising Stars of Cancer Metabolism Symposium in 2021. Christy's also taking on many leadership roles, both in the field and at the Salk Institute, and is also very active in DEI initiatives, including promotion and mentoring of junior and URM students and faculty. And obviously this is an area where we're all trying to do a better job these days. So Christy is also really leading the way there as well. So without any more then, I will hand over to Christy, and excited to hear what you have to say. Thanks very much, Christy. Looking forward to it.
Thank you so much. I will go ahead and share my screen. Well, thank you for that kind introduction. And as you just mentioned, I am a sort of brand new assistant professor at the Salk Institute, started my lab about 6 or 7 months ago now. And so today I'll talk a lot about the work that I did as a postdoc that launched my lab. And then at the end, I'll circle back to some of the things we're really excited about as we launch our own program here. So the title of my talk today is Mechanisms to Circumvent Autophagy Inhibition in Cancer. So as many of you already know, autophagy, there are 3 flavors of it. I'm most interested in the most well-studied, which is macroautophagy. And it's a cellular recycling process that degrades damaged organelles and proteins, and in the process generates building blocks like nucleotides. and other things that can go on to fuel metabolism and cell growth. And it is a multi-step process that involves the formation of a double membrane autophagosome structure here that can go on to fuse with lysosomes via the snare protein like STX17. And once that fusion happens, that leads to the breakdown of the material. There's over 20 core proteins involved in this process, but I'll just highlight the LC3 conjugation step, which is critical for the formation of autophagosomes. So this is where phosphatidylethanolamine is conjugated to LC3-1. And this is mediated by the E1-like enzyme ATG7. And so LC3 conjugation and therefore ATG7 is absolutely critical for the formation of autophagosomes and for the entire pathway or process to happen. So autophagy is important in a lot of different disease states and pathologies, but we are most interested in the roles of autophagy in cancer cells. And there's been a lot of work that's been done by many in the field, including Jay here today. But a lot of this work has led to over 60 clinical trials that are now moving forward targeting autophagy. And this is really based off of cornerstone studies by Eileen White and Alec Kimmelman, as well as many others that have shown that targeting autophagy in established tumors can reduce tumor growth in mouse models and in cell lines. And so now this is a very long list that I know is too small to read of the current clinical trials that have recently completed or are still ongoing. And the point of showing this is just to highlight the diversity of these clinical trials, which are really moving forward in, I'd say, your favorite tumor type. All using late-stage autophagy inhibitors like hydroxychloroquine or chloroquine, which target the lysosome. And initial trials have been somewhat promising. So this is in glioblastoma with a group of patients treated with chloroquine, and you can see that the patients treated are doing better than the control group, suggesting that this may be a viable therapeutic moving forward, which is really exciting for the field. But I would also argue that a lot of the ongoing trials are showing inherent resistance, meaning large groups of patients are not responding at all to autophagy inhibition. And then even more interesting is the second bullet point, where the clinical trials are seeing patients that do initially respond, but then they start growing on therapy, right? So this, of course, would be acquired resistance. And again, while I think the field in general is really excited about these clinical trials moving forward, to me, this is a bit of a red flag, which is, can cancer cells actually adapt to autophagy inhibition? And could there be mechanisms of resistance that we really should be studying to try to understand this as these clinical trials are moving forward? And so to try to build a system in order to study this, I wanted to design a technique that would allow us to really understand with temporal resolution how essential this process is. And so during my postdoc, I created an assay that's a unique flavor of CRISPR-Cas9 that's coupled with live cell imaging, and I call it LC-CRISPR. And so for this assay, I'll walk you through what we do. We start off with cells that are double positive with an M-cherry NLS or nuclear localization signal, as well as a GFP NLS. And so our cells start off yellow. And then in vitro, we can build our ribonucleic particles, which include a recombinant Cas9 protein, as well as an in vitro transcribed guide RNA. And then we can use a lipid-based transfection to transfect this in. And importantly, in our system, we use guides that target GFP as well as target our gene of interest. And so we can then use loss of GFP as a surrogate for loss of our gene of interest. So we can couple this with live cell imaging, which then allows us to monitor the effects on viability in real time. So immediately after editing, we're not waiting weeks to grow these clones out, and we're not waiting even days to clear antibiotic selection. And so then within the same well, we can measure the viability or the growth of the cells that are edited and have lost GFP, and therefore are red, versus the cells that are still double positive and are yellow. And we can do this in a semi-high throughput manner in a 96 well plate, where in an arrayed fashion. So every well is getting a different guide RNA. And then on every plate, we have control guides that are targeting either known essential or known non-essential genes to compare. And then what's great about this is that it's not an endpoint assay, so we can actually flow sort these out based off of fluorescence and assess things like gene expression. So using this assay, we were able to show that within at least 7 days immediately after editing, there are a lot of cancer cells that are highly dependent on autophagy. So when we knock out autophagy genes, they look sort of like this red line where the cells can't grow if they have the autophagy genes knocked out. And so sort of zooming in on some of that data here, we're looking at a cell line that we have identified as highly dependent on autophagy. So here we're looking at the growth of the M-cherry positive, but the GFP negative population. So just the edited population. And we're comparing knockout of ATG7, again, critical for the formation of autophagosomes. And we're comparing that to knockout of a known essential gene like elongation factor 2 versus a known non-essential gene, the tumor suppressor P10. And so you can see that here, again, looking immediately after editing for about 7 days out, we can see that knocking out ATG7 looks identical to when we knock out a known essential gene, just drastically different than if we knock out a non-essential gene. So indicating these cells are highly dependent on ATG7 for survival. But then what we did was something really interesting, and we actually left the cells in the dish, even though they all looked pretty mad, pretty dead. We kept replenishing their nutrients, and eventually the cells started to grow again. And eventually we could grow these out and clone them out, and we could identify ATG7 knockout clones that were completely deficient for ATG7, and they have no LC3 conjugation, and they have no canonical autophagy. We can see an accumulation of the autophagy substrate P62. So these cells took a very long time to grow out, and they were quite rare within the population. But of course, the question was, these cells that were derived from highly autophagy-dependent cells, how happy or functional could they be without ATG7? And what was surprising is that these ATG7 knockout clones could grow identical to the wild-type cells from which they were derived. This is true under full nutrient conditions, but also if we starve the cells, which is a context that should make them extremely dependent on autophagy. So again, these cells are derived from highly autophagy-dependent cells, but now they've switched to become autophagy independent. And we did a lot of studies to really prove that, and I'll just show one here where we're looking at apoptosis using caspase 3/7 activation as a readout here. And now we're treating with the pharmacological agent chloroquine. And you can see the wild type cells are undergoing lots of apoptosis, which makes sense. These are an autophagy-dependent cell line. But our ATG7 knockout cells are really resistant to this. This was true in vitro as well as in vivo. If we xenograph in the tumors, the wild type tumors are sensitive to chloroquine, but our ATG7 knockout cells are now completely autophagy independent and resistant to hydroxychloroquine. So of course, these cells have switched from being autophagy dependent to now being autophagy independent. So the million-dollar question is, how are they doing that? And so we went after two questions, which is how are they maintaining protein homeostasis and mitochondrial homeostasis. And we went over after these two questions because we know autophagy is really critical for these two processes. So really quickly looking at the first question, and I'll just summarize this because it's been published quite some time ago now, and I'll just summarize it in graphical abstract form, looking at how these cells maintain protein homeostasis. And what we showed was that when we block autophagy in these highly autophagy-dependent cells, they actually have a decrease in proteasome-mediated degradation, and that's because they're accumulating damaged proteasomes, which would normally be degraded through autophagy. And in order to circumvent this, they have activated NRF2, and they do this in a P62 keep one dependent mechanism. And they activate NRF2 in order to build more functional proteasomes. And we showed that NRF2 directly transcribes as a master transcriptional regulator, directly transcribes to build more functional proteasomes. And the one piece of data that I will show that I think really highlights this the best is here, where we're treating with bortezomib, which is a proteasome inhibitor, and looking at apoptosis as a readout, or using caspase 3/7 as a readout for apoptosis. So here, if we look at the solid lines, the blue lines are our ATG7 knockout cells. They're slightly more sensitive to bortezomib because they don't have fully functional proteasomes. But in order to get around this lack of fully functional proteasomes, they've activated NRF2. And so now if we knock down NRF2, they become extremely sensitive to bortezomib. Again, indicating they're using this mechanism as sort of a workaround to circumvent. Okay, so now moving on to the second question, which is more recently, we looked at how are these cells maintaining mitochondrial homeostasis? And I think this is a really interesting question because we know that only way to really degrade an intact mitochondria is through selective autophagic degradation of mitochondria, which is called mitophagy. And so, you know, if our cells are not making autophagosomes, they're not doing mitophagy, how are they maintaining mitochondrial homeostasis? So we first asked, well, are they doing canonical mitophagy? And for this, we used pH-sensitive probes, which were originally created by Ian Ganley's lab, which takes advantage of the pH-stable M-cherry fluorophore, as well as the pH-sensitive GFP fluorophore, all in tandem and localized to the mitochondrial outer membrane with a FIS1 sequence. And so if our mitochondria are in a neutral environment, then both fluorophores would express and our mitochondria would look yellow. But if our mitochondria are in a low pH environment, then the GFP is quenched. And then we can quantify the ratio of these two fluorophores using flow cytometry. And so when we do that, and we're looking again at the ratio of M cherry to GFP, we can see if we starve the cells, which is a context known to induce canonical mitophagy, we can see a lot of mitochondria degradation, a lot of mitochondria getting into a lysosome here in our wild type cells, but we don't see any canonical mitophagy in our autophagy deficient cells. Again, these cells were originally derived from highly autophagy dependent cells. So if they're not doing any canonical mitophagy, they can't degrade damaged mitochondria. How healthy and functional could their mitochondria be? I think the most telling experiment we did actually was when we forced the cells to grow in galactose, which is a sugar that cannot be easily metabolized by glycolysis, forces the cells to use their mitochondrial respiration pathways. And in fact, we can see that these ATG7 knockout cells can grow identical to the wild type cells here. So again, shows that at least when forced to, they seem to be able to use their mitochondria just fine. And sorry, put the slide in twice, but again, here we're showing if we look at oxygen consumption rates using seahorse assays, again, we saw no change in our ATG7 knockout cells. Again, indicating these cells seem to have pretty healthy mitochondria. So taking a closer look at their mitochondria by EM, here looking at our ATG7 knockout cells, the mitochondria look pretty happy and healthy. We can see nice intact Christie here. But what I also hope you can appreciate is that compared to the wild type cells, their mitochondria are really elongated. And so we went on to show that the cells have increased mitochondrial fusion, and we could show the ATG7 knockout cells have a reduction in the number of cells that have this more fragmented phenotype and an increase in the number of cells that have this more hyperfused phenotype. So a little bit more about mitochondrial dynamics. It's really a yin and yang between fission and fusion, which is all regulated by a set of GTPases, where fission is regulated by DRP1 and fusion is regulated by these mitofusins. And so we could show that our cells had a lot more active mitochondrial fusion, but we wanted to ask if they were actually dependent on this. So to block mitochondrial fusion, we expressed a dominant negative form of mitofusin 1 with a mutation in the GTPase domain. And here looking at adjacent cells that are either not expressing the dominant negative and still have this nice elongated mitochondrial phenotype versus cells that are expressing the dominant negative, which is MIC tagged here, we can see this more fragmented phenotype. And so using this construct to block mitochondrial fusion, we saw really no effect on viability in our wild type cells, but our ATG7 knockout cells showed drastic reduction in growth when we block mitochondrial fusion. And this really makes sense because fusion has been shown to be a cytoprotective process. It allows cells to take bits and pieces of damaged mitochondria and sort of fuse it to the entire mitochondrial network, keeping kind of the whole network more intact. And so we really showed that these cells were using more mitochondrial fusion to maintain mitochondrial homeostasis, which is great, but I think still a big question remained, which is, can these cells actually degrade damaged mitochondria? And if so, how are they doing that, right? Or are they just gonna go on to keep undergoing mitochondrial fusion forever? So to sort of get at that question, actually, we came across these very fascinating mitochondrial derived vesicles. And I must admit, when we first were working on these studies, I had never heard of these MDVs. And we learned about them at a Keystone meeting. And I sort of maintained this philosophy that all trainees should go to as many like Keystone and Gordon meetings as you possibly can. I feel like that's where all things are learned. But anyways, we came across these very fascinating mitochondrial derived vesicles, which were originally coined by Heidi McBride's lab in Quebec. But they're fascinating because if you look at all forms of mitophagy, they really involve LC3 mediated autophagosomes here, whether they be PARC independent or independent, they really all are LC3 mediated. But these mitochondrial derived vesicles are tiny vesicles that can sort of shed from the mitochondrial membrane and then go directly to lysosomes. And they do not go through autophagosomes. And so of course, we were interested, could our cells be creating some of these mitochondrial derived vesicles? And so combing through our electron micrographs, we thought we could maybe identify some of these vesicles, but it's really hard to quantify through EM. So instead, we turned to immunofluorescent and confocal microscopy. And so here we took advantage of an assay, again, that Heidi had originally used to identify these MDVs, which is based off of the concept that these MDVs are they're highly cargo selective. And so if we co-stain for an inner mitochondrial protein like pyruvate dehydrogenase, as well as an outer mitochondrial protein like TOM20, all of the mitochondria that are intact will co-stain for both. If we zoom in on this sort of region of interest here, and we draw an ROI, we can see a intact mitochondria that is both PDH positive and TOM20 positive, but these MDVs, which are cargo selective, are either PDH positive or they're TOM20 positive. And so if we look across this ROI here, starting from the left, we see our intact mitochondria, and then we come across just a PDH positive mitochondria derived vesicle here. And then all the way here on the right side, we can see just a TOM20 positive MDV. And so we can use this assay to count or quantify these MDVs. And by doing this, we saw that our autophagy deficient cells, again, derived from highly autophagy dependent cells, but have sort of switched to become autophagy independent. And now they are making more of these different types of mitochondrial derived vesicles. And so if we look at the total number of MDVs, they really have a significant increase. But I think what's most interesting is that if we treat the cells with any sort of mitochondrial toxin or mitochondrial damage, so here we're treating with DFP, which is an iron chelator, we can see that they increase the amount of MDVs they're making even more, suggesting that they're using this sort of alternative pathway to still degrade mitochondria when it's damaged. Okay, so now that we could count and quantify these, I actually wanted to ask, can we monitor their flux through lysosomes? And so I wanted to take advantage again of this FIS1 tandem construct, which again has M-cherry, which is pH stable, and GFP, which is pH sensitive, all localized to the outer mitochondrial membrane. And so in cells that have stable expression of this construct, we stained for TOM20 and PDH and could identify some of these mitochondrial derived vesicles. And then we pseudo-colored here the M-cherry blue and the GFP white. And if we overlay all of these colors, which I think is the easiest, and we draw an ROI, starting at the bottom here, which is corresponding to the left side of the graphs here, we come across a outer derived, or TOM20 positive, but PDH negative mitochondrial derived vesicle, which does label with the FIS1 tandem, which again makes sense. The FIS1 tandem is supposed to be localized to the outer mitochondrial membrane. And then we come across the top here. So moving to the right, we see an intact mitochondria, which is TOM20 positive and PDH positive, and then of course also has our FIS1 tandem. But then as we come across to the top, which is the right side, we come to a TOM20 negative, PDH positive derived mitochondrial vesicle, but it does not label with the FIS1 tandem. So this was great now because it meant that we have a construct that can specifically label different types of these MDVs, and it's a pH sensitive construct that we can use these really sensitive quantitative flow cytometry assays to measure or monitor their flux. And so now we use the flow cytometry to measure the flux here. And so in our wild type cells, where we treat with different types of mitochondrial toxins like DFP, which is an iron chelator, or CCCP, which is a mitochondrial uncoupler, we see lots of mitochondria that are getting into a lysosome in our wild type cells. But now, of course, in our wild type cells, we have canonical mitophagy happening as well. But if you remember, I showed that our ATG7 knockout cells were not doing any canonical mitophagy. But now when we treat with these mitochondrial toxins, we do see that they can still get mitochondria into lysosomes. So presumably through these mitochondrial derived vesicles. And so what we're showing here, kind of graphical abstract form, is that if we have these TOM20 positive MDVs, we can show with these FIS1 tandem constructs, we can really quantify these MDVs. And we also think we have pH sensitive constructs that we can localize more to the inner mitochondrial membrane, and which can preferentially label the PDH positive MDVs. And we can now use these different assays to really quantify and understand the flux of these constructs. So, but next, of course, the question is, well, can we manipulate these MDVs to ask, are they necessary or relevant for sort of the physiology or survival of cancer cells? And so to manipulate these MDVs, we knock down sorting nexin 9, which has previously been shown to be important in MDVs, but in macrophages and never has really been studied in the context of cancer cells. So we showed that if we knock down SNX9, we did see a decrease in the number of MDVs. And so we could ask, is it important for, are MDVs important for survival? And so here we're looking at apoptosis, again, using caspase-3/7 as a readout. If you look at the gray lines here, this is knocking down SNX9 in the wild type cells. So we do see some apoptosis happening. But if we knock down SNX9 in the autophagy deficient cells, we see drastically more apoptosis happening, again, indicating that these cells have an acquired dependency on SNX9 mediated MDVs. And then I think a more telling experiment perhaps is when we treat with CCCP, which again is a mitochondrial toxin, it's a mitochondrial uncoupler. If we look at the solid line, the solid red line compared to the solid gray line, that shows that our ATG7 knockout cells are slightly more sensitive. They're undergoing slightly more apoptosis when we treat with CCCP, again, perhaps because they don't have perfect mitochondria degradation going on. But in order to still attempt to degrade mitochondria, they're creating MDVs. And so if we knock down MDVs by knocking down SNX9, now they become drastically more sensitive to CCCP, again, indicating they're using this process to circumvent sort of the canonical pathway. But of course, here we're looking at overall apoptosis, and SNX9 does a lot of things besides mediating MDVs. So how do we know this is mitochondrial specific? So we turn to our oxygen consumption rate assays using seahorse here, knocking down SNX9. And we saw really no effect in the wild type cells, but we saw a dramatic effect in our ATG7 knockout cells. Again, I think really solidifying the hypothesis that these autophagy deficient cells are using SNX9 mediated mitochondrial derived vesicles in order to degrade mitochondria when the canonical autophagy or mitophagy pathway is not an option. Okay, and so putting that all together in a model, what we've shown is that if we block autophagy in highly autophagy dependent cells, the vast majority of the population die, but there seem to be these rare cells that can survive and adapt and then go on to recover, such that the recovered population can grow actually identical to the wild type cells from which they were derived. And we showed that these cells have now switched to become autophagy independent, and they can grow identical to the wild type cells when we treat with an autophagy inhibitor. But in the process, they've now acquired novel susceptibilities. And so now they're more sensitive to when we block the proteasome or when we manipulate NRF2. And what I showed today is they're really more sensitive to when we block mitochondrial fusion or mitochondrial derived vesicles. And so what are the mechanisms that have led to these novel susceptibilities? If we sort of zoom in on our healthy cancer cells here, we have intact autophagy, which has helped maintaining functional proteasomes, and we've got basal amounts of NRF2. But when we we have happy and healthy mitochondria that when damaged can be degraded by mitophagy. But when we knock down or knock out the autophagy pathway, we start accumulating damaged proteasomes and some pretty pissed off mitochondria hanging around. But as these cells recover, they activate NRF2, which helps build more functional proteasomes. And they start doing more mitochondrial fusion to help maintain overall mitochondrial homeostasis. And they are creating more of these mitochondrial derived vesicles to still degrade mitochondrial material. So if we swap out the model for what I think are still the most interesting outstanding questions, which are the things that we're really interested now in pursuing in my own lab, is still really interested in which tumors are actually sensitive to autophagy inhibition. Can we identify biomarkers that would help us identify the ideal patient populations to be treated with autophagy inhibitors? And we're really moving into pancreatic cancer with these studies and using more physiologically relevant PDAC organoid models to study these questions. We're also really interested in the idea of whether or not this is truly an adaptation, or is it just due to low levels of heterogeneity? And is this a clonal variation versus an acquired adaptation? And we're using single cell sequencing barcoded technologies to answer that question. Of course, we're really interested in the differentially essential genes in the recovered population, as these would be ideal candidates for combination therapies with autophagy inhibitors. And we're collaborating with Ruben Shaw's lab to create genetically engineered PDAC and lung models to ask these questions. We're also really interested in how fast these cells can adapt. We have data to suggest it might be quite rapid, actually. And so we're using optogenetic techniques and eventually moving into model organisms to analyze these questions. And then, of course, we're fascinated by these mitochondrial-derived vesicles. Again, they're highly understudied. There's less than like 10 publications on them. And so really interested in what's inside of them, where are they going, how are they trafficked there? There's just so many questions still to answer. And so we're doing some unbiased genome-wide screens, as well as super resolution microscopy technologies and techniques to get at some of these questions. And so with that, I will acknowledge my postdoc lab, which a lot of the work I talked about today was done during my postdoc. But of course, I have said goodbye to the beautiful Rocky Mountains and have moved here to Salk. And so also want to thank my current lab here at Salk. And we are, as I said, a brand new lab and aggressively growing and hiring. And so hiring postdocs and technicians and students. So please check us out, any of our social media or the lab website here. Would love to talk more to trainees about joining our group. And with that, I will take any questions.
Wow, that was an excellent talk. I have to tell you, we already have a lot of questions, and I don't know how many we'll get through before we have to go to Jayanta's talk. I'll start with a question that I was also interested in that Peter asked, which was, so you have these MDVs, and these MDVs potentially fuse to become MVPs. How many of them get targeted to the lysosome versus is there a role for extracellular secretion here as well? What's the balance?
Yeah, that's a fantastic question. And the truth is we really don't know. We do know that the trafficking of these vesicles is Rab7 mediated, meaning that they are going through late endosomes, right? Which would mean they would be prime candidates to become exosomes. But we really don't know sort of what is dictating whether they actually traffic to lysosomes or if they are potentially becoming exosomes and what they could be doing if they are becoming exosomes. But I do think some of these techniques that we now have and these tools that we have, we can really use this to ask this question, actually track, be able to watch this with live cell imaging now and see where these MDVs are going. But to date, we really don't know.
Yeah, fair enough. And another interesting question, which goes back to your clonal selection, like what is the cause of this autophagy dependence? Jayanta, actually our other speaker today, had brought up the idea of, have you tried doing just partial blockade of ATG7? So in other words, this I'm paraphrasing, do you think that there's maybe a population that has lower levels of ATG7, which are more easily able to adapt? But that second part is mine. The first part was Jayanta's.
Yeah. I mean, that's a really interesting question and probably the most physiologically relevant question, right? Because we're never going to knock out the entire pathway in a patient. Yeah. We're going to be partially targeting the pathway. And so, we could obviously look at that maybe in with our CRISPR screens, we can look at it with our HET lines, right? But I think probably one of the best ways is to actually ask, do these same mechanisms happen if we do it with pharmacological agents, right? So we're working on those studies right now, which is if we can make cells resistant to chloroquine, for example, do they use these same pathways? And could we combine therapies to target these with hydroxychloroquine? And so we're tackling those questions right now.
Okay. Zeb had a good question as well. How does this increase in MDV? I know you said there's only been 10 publications, so this might not have been covered, but does increase in these MDVs have implications for tumor microenvironment, immune surveillance?
Yeah. Yes. Great question. So again, not a lot has been done, especially in the cancer context, but I will say that Heidi McBride, who originally identified these, did have a paper come out now 4 or 5 years ago now, showing that these MDVs can actually be important for antigen presentation, at least in macrophages, but it does beg the question, could cancer cells be using these to sort of present cancer antigens in some sense? So those are questions that, I think with collaborations with some great immunologists here on the Mesa, we could start to tackle some of that, but it really hasn't been studied in the cancer context. But again, because these could become exosomes, there's definitely the chance. I mean, I would hypothesize that they are playing a role in the tumor microenvironment and that they are affecting sort of the immune properties of the tumor, but we haven't been able to study that quite yet.
Really exciting future directions. What just a last question, and then I'm going to let Jayanta give his talk, and then I'll come back. There's a couple of questions that I'm saving for later that I think would benefit from both of you giving your perspectives on it. But this is a good question in terms of more in the weeds of your talk. Have you looked at the ER UPR regulation of mitochondrial dynamics, homeostasis, and how does that go with these MDVs that you're finding?
Yeah, great question. Great question. We really have not tackled the ER or UPR mechanisms as of yet, and that's just hasn't really been our expertise, but always love to collaborate to ask some of those questions, because I think we would be naive to think that this is happening in a vacuum. Obviously, there's lots of mitochondrial and ER contact sites, and where are these MDVs shedding? Does the ER play a role in that in terms of contact sites? All sorts of questions that, would love to pursue.
So that's anyone who's doing mitochondrial dynamics, email Christina. All right, I'm going to let Jayanta do his talk, and Peter will introduce him, and then we'll come back at the end for more questions.
Great, Christy, that was excellent. Thank you very much. And I'm looking forward to meeting you this afternoon. Great, so I'm going to introduce Jayanta Debnath now. Jay has been an independent investigator at UCSF since 2005. And he's now a distinguished professor and the chair of pathology at UCSF School of Medicine. So his lab has a long-standing interest in the role of autophagy in survival of epithelial cells and transformation of epithelial cells, and also tumor metastasis in vivo, using in vivo models, and ultimately wants to understand these mechanisms so that he can develop novel therapies to target autophagy in cancer. Another area that he, more recently that I think he's got, he's kind of leading the way in, a pioneer in this area, is the role of autophagy in secretion. I think sometimes known as secretory autophagy. Okay, and so this is describes what I think is still, quite poorly understood, but the role of non-canonical functions of autophagy regulators in secretion of components from cells as opposed to simple degradation. So that's kind of a new and exciting area where Jay is leading the way. He has many awards, most, a couple of recent ones. In 2021, he was awarded the Outstanding Investigator Award from the American Society of Investigative Pathology. And in 2013, a Waxman Investigator Award, and many others before and between those awards. So yeah, I'm really looking forward to Jay's talk and also meeting Jay later this afternoon. So just remind you all, if you could put your questions into the Q&A, please. Great. Over to you, Jay. Thank you very much for coming. We're really looking forward to your talk. Thank you.
Yeah, thank you for having me, and this has been a great symposium already. It's great to see Christie's latest stuff. So as was pointed out, I have been working in this field kind of since the dawn of time of this field, and there's been, as Christy pointed out, a major interest in actually targeting this pathway for benefit in cancer patients. And was already discussed, the primary modality we've been doing this by is actually shown here in this red box, is that we've attempted to inhibit the late stages of autophagy using antimalarials such as hydroxychloroquine. What's important to remember is that all of these agents have broader effects on the lysosome, so they would affect other pathways such as macropinocytosis, endocytosis, and phagocytosis, and so they have broader effects, but nevertheless, they've been utilized for their effects on autophagy inhibition. And where we stand today, and I think Christy actually introduced this very well, is that we know that certain tumors, most notably that have very highly metabolically driving mutations, such as RAS, RAF, LKB1, seem to be highly sensitive to autophagy inhibition. We also know through these trials that they've broached the promise of this as a therapeutic strategy, but we've also seen mainly pharmacodynamic limitations of their use in humans. As Christy very nicely delineated, we know that there are developing mechanisms of acquired resistance to autophagy inhibition, and also that autophagy inhibition isn't actually just a simple panacea, in the sense that we have seen that this same pathway that kind of inhibits primary tumor growth seems to paradoxically increase metastatic recurrence and progression. And my lab actually showed this in a paper a couple of years ago, where we were able to see significant regression of primary tumor growth and this increase in metastatic outgrowth due to the accumulation of a cargo receptor called NBR1. So these results overall, highlight the complexity of autophagy in using it therapeutically in cancer. And they also really highlight two fundamental near-term needs, and that's we need more chemical matter to kind of specifically stimulate and inhibit autophagy and figure out the context where we need to be applying this. And we also need better strategies to non-invasively monitor this process, especially in our patients, where we actually manipulate this pathway either up or down. The other thing that is more of a biological question is that we've known that targeting autophagy, whether it being cancer or any disease, it has broader effects on the overall host, and it influences pathways other than lysosomal catabolism. And what I want to focus today on is actually these non-cell autonomous contributions of autophagy in cancer progression. And this is a newer concept in the field because for many, many years, for almost 2 decades, we really took this tack is that, how is autophagy killing or not killing a cancer cell? Really I've come to realize that when you inhibit autophagy in a single cell or a single compartment, it has influence on other compartments. And of course, this has implications for the tumor microenvironment or any type of microenvironment where you're actually targeting this pathway. And this concept was first actually kind of appreciated through the concept of metabolite exchange. There were a series of very seminal papers from the Kimmelman lab, Torek Ruskin's lab, as well as Arlene White's lab, suggesting that when you inhibit autophagy in the host, there's certain amino acids that are not being secreted or released, and those happen to be something that is necessary for tumor cells to sustain and their growth and in a variety of organisms, both fly as well as in mice. And so the main thought was that how autophagy was used as a communication mechanism has been that if you inhibit autophagy in one pathway, you have a metabolic defect in that pathway. That no longer can now be utilized by other cells due to the transfer of those metabolites. What we have been focused is a variation of that, is in the sense that autophagy is actually more directly involved in communication. And I actually just show a figure from a review that we wrote with Jose Torres, who's with you, as well as Melina Hansen, discussing actually the various aspects of the autophagy machinery in both degradative and secretory pathways. And the concept of secretory autophagy was originally described in yeast by Suresh Subramani, as well as Vivek Malhotra, and really focused around proteins that are unconventionally secreted. And it was actually extended into mammalian cells by Voya Deratik's lab, as well as Randy Sheckman's lab. And what we've come to appreciate is that actually autophagy influences a variety of mechanisms in which, instead of actually using the machinery to send things to the lysosome, it is actually being used as an alternative way to send things outside of a cell. What's important to remember is that these are multiple different secretory pathways, and my own lab has actually uncovered at least 2 of these, both of which actually involve intermediates that involve extracellular vesicles. We published the role of LC3 by non-canonical pathway in extracellular vesicle loading a couple of years ago, and recently just sent the paper back of looking at secretory autophagy and what is being secreted via EV populations during lysosomal inhibition. Most of this work has been pretty fundamental cell biology and really in the context of age-related neurodegeneration. Since this is a focus on cancer, we've always been interested in this related question, is that how does secretory autophagy contribute to intracellular communication during cancer progression? And so a few years ago, we set out to answer this question by looking at a single cell type, a highly secretory cell type in the tumor microenvironment called the stromal fibroblast. And we asked a simple question whether if you deleted or inhibited autophagy in a single cell in vivo, would it actually promote or suppress tumor in the context in which the tumor cells were completely autophagic competent. So we deleted this using a fibroblast-specific Cre driver called FSP Cre. And what we found is if you inhibit two different ATGs or delete them only in the fibroblasts of a mammary fat pad in an autochthonous mammary cancer model driven by polyamidyl T, this is your typical polyamidyl T growth over 80 to 100 days. It's significantly attenuated by deleting either of these only in the fibroblast. And so this was actually a telling experiment for us because when you do this same experiment in tumor cells, although you do have an overall inhibition and growth, it's highly context dependent. In this case, actually, it was pretty much night and day, and it was a very striking phenotype for us. And so we set out to try to understand why this was occurring, in this, simply by deleting autophagy in a single population in the host. And we really became interested in a pathological phenotype called desmoplasia. Desmoplasia is the proliferation and the activation of fibroblasts in the context of a growing tumor. Many tumors actually, many solid tumors exhibit this, including breast cancer, which I show here. But probably the poster child for this is actually pancreatic ductal adenocarcinoma, which actually, if you look at this pathologically, most of it is desmoplastic stroma rather than, rather than the tumor cell compartment itself. This also has an immune component to it. It's immune cell rich, both innate and adaptive components. And this is invariably, if you do pretty much any epidemiological study, associated with reduced patient survival and with increased metastatic disease across all tumors. There really isn't any place, at least in humans, where desmoplastic response is not a predictor of poor prognosis. And so if you could think of mechanisms that short circuit that desmoplastic response, that would obviously be of benefit in the context of tumor progression. So I just want to point out to you that the model we are using exhibits significant desmoplasia, both in terms of fibroblast infiltration, as well as infiltration of hematopoietic cells marked by CD45. And when we looked at when you deleted autophagy in this context, those desmoplastic components seem to attenuate. So this is a trichrome stain. The blue is collagen. It is significantly decreased in the context of autophagy dilution only in the fibroblasts. And then at the same time, we see infiltration of various populations, such as macrophages, as well as lymphocytic populations into these tumors is significantly reduced. And so we started to ask, why is this happening? Why is the reduction of autophagy in a single cell type starting to attenuate this desmoplastic response? And our first thought was that these fibroblasts were just non-specifically sick. Autophagy is a major adaptation pathway. A solid tumor is a stressful situation. They're just not really doing well. That actually turned out not, as far as we can tell, really not to be true. So if we do metabolic profiling, I'm showing your seahorse assays, you actually see a slightly better metabolism in autophagy deficient fibroblasts compared to autophagy competent fibroblasts. So these would be either deleted for ATG5 or ATG12, and you just look at oxygen consumption. And we've done also steady state metabolomic profiling and really have never seen any kind of smoking gun, at least in fibroblasts. And this contrasts really greatly with what we see if we did this same experiment, for example, in a tumor cell or a cell that was transformed with an oncogene, such as RAS or RAF. We also find that you can activate these fibroblasts perfectly. So the best way to activate a primary fibroblast is simply to grow it over time on tissue culture plastic. They activate all the canonical markers such as FAP, smooth muscle actin, FSP, fibronectin, and many others. And then if you look at a typical fibrogenic program such as TGF-beta, the signaling events seem to be intact regardless of whether you have autophagy as competent cells or deleted due to the loss of ATG5 or ATG12. So what is going on? So it turns out that we had already shown that autophagy affects secretion of certain cytokines using three-dimensional culture models. We published that in Cancer Discovery almost 10 years ago now. And many of those same cytokines were actually showing up as deficiencies in fibroblasts lacking either ATG5 or ATG12. But it's not a broad-based defect in secretion once again. So something like IL-6 is secreted at reduced levels. On the other hand, TGF-beta is secreted actually perfectly fine. And so one of the things that we did consider is whether this was an unfolded protein response, and we really have never seen any evidence that the secretory pathway is being negatively affected by the kind of activation of an unfolded or ER stress response in these fibroblasts. So we did some cytokine profiling, and there are very specific cytokines that are actually attenuating, but most of them are those that support a desmoplastic microenvironment. At the same time, we actually turned our attention to kind of the real day job of a fibroblast, which is actually secreting ECM components, things such as collagen or fibronectin and so on and so forth. And we actually really were inspired by work in the developmental biology field that showed that actually autophagy is critical for collagen 2 secretion and proteostasis, particularly during bone growth. And so we asked, these same fibroblasts in mammary gland or in other places such as the lung or the liver, their day job is secreting things such as collagen 1 or type 1 collagen. And we asked if this is the case as well for these types of cells. And by the way, this original work in bone growth was actually pioneered by Carmine Septembre and his colleagues, and has been a really beautiful story in the dev bio field. But we asked if this was actually going on in fibroblasts. We first looked at this in vitro. So the problem with in vitro is that the collagen secretion response is pretty wimpy with fibroblasts, especially primary cells. But nevertheless, we do see that the loss of ATGs reduces the amount of collagen you see being put into the media. And if you acutely inhibit autophagy with a drug such as spoutin, you see very similar phenotypes. At the same time, we saw the proteostatic defects that Carmine's lab reported. So these ATG5 and ATG12 fibroblasts actually seem to have defects in collagen or type 1 collagen proteostasis. And so just to take you very briefly through these assays, a normal fibroblast, you get a subpopulation of collagen that goes to the lysosome, and these co-localization events, which is basically LAMP2 and collagen, are actually now pseudocolored black in these kind of ink blot squares that you see below the double labeled immunofluorescence. And so you can see in a normal fibroblast, a lot of collagen is actually being sent to the lysosome, and you can actually trap this even more with an inhibitor of lysosomal function, such as vancomycin. Now, if you take away either ATG12 or ATG5 in mammary fibroblasts, these events are significantly attenuated. And so this really is an analogous to what was seen in the bone field and bone kind of stromal cells, in a sense that you get defects in collagen proteostasis, which has been proposed to lead to a secondary effect of a defect in collagen secretion. And in this case, we would argue that it's type 1 collagen secretion. So I told you that the collagen secretion defect in vitro was pretty wimpy. And so we decided to actually look at this now in vivo. And to do that, we actually took advantage of the mammary gland, where you can now put in defined elements. And so we essentially just adapted a technique by Bob Diome and more recently used by Sharp Hooperwasser to actually just prime fibroblasts or mammary fat pads with fibroblasts. But in this case, the fibroblasts were now genetically defined, either wild type or autophagy deficient. These fibroblasts, regardless of whether they're wild type or autophagy deficient, reconstitute at equivalent rates. We don't really see any differences in cell death, in cell number, in cell proliferation. However, if you look at the amount of collagen, either by H&E or by trichrome staining, the amount of collagen that is deposited by these fibroblasts in vivo is significantly attenuated. And we can quantify this with an analogous stain to trichrome called P-Posterius red, where you can use the refraction index as a fluorescent marker and quantify that. And you can see here that autophagy deficient collagen deposition is significantly attenuated to fibroblasts that are autophagy competent within the context of these primed mammary fat pads. So what does this mean for that by the fat pad? Well, in collaboration with Val Weaver's lab, we've shown that this actually affects the stiffness of the fat pad. Fat pads reconstituted with autophagy competent fibroblasts have a much stiffer matrix that compared to those that are autophagy deficient, either due to the lack of ATG12 or ATG5. We can also model this in vitro. If you in vitro introduce autophagy deficient cells, either 12 or 5, they have significant impairments in terms of collagen contraction. We don't know if this is actually due to a secretory function or a actual contractile function of the fibroblasts themselves, but nevertheless, this also contributes to this, this kind of decrease in stiffness phenotype that we're seeing in vivo. And so many in the field, most notably Val Weaver, have proposed that this stiffness is a hallmark of desmoplasia and is thought to be tumor promoting because it promotes the ability of the tumor cell compartment to proliferate due to engaging specific integrin-mediated events. So we asked what this would mean for tumor growth. So we took this priming assay that I told you. We've generated this kind of primed mammary fat pad, either competent or incompetent due to the cells we put in. And now we put in, a few weeks later, an autophagy competent polymiddle T tumor cell. So the tumor cell can do all the autophagy we want. And so this is an analogy to our autochthonous models that we do, but it's actually very specific, very cleanly titrated, and and so on and so forth. And we get essentially the same result. You do not grow tumors when you are in the context of an autophagy deficient fibroblast, and these mice survive indefinitely, where those grown in the context of wild type fibroblasts succumb over time. We can actually do this in a completely different way. We can co-mix tumor cells with either an autophagy competent fibroblast or an autophagy deficient fibroblast. And once again, we see exactly the same result, a reduction in kind of tumor growth and volume over time. So what is going on? So we think that this is a multifactorial effect of the stiff collagen matrix that has been developed. And one piece of evidence of this is if you look at stiff matrices, either in the fibroblast plugs, as I show here, or in the tumors, they have reduced microvessel density that is quantified here. So a fibroblast environment that is composed of wild type cells has significantly more kind of vascular density compared to that has been reconstituted using ATG12 deficient things. And I just want to remind you is that one of the most potent in vitro defects that we see is IL-6, which is both a potent fibrogenic cytokine, but also a potent kind of neoangiogenic cytokine. So we at least hypothesize that it is playing a role, but we've not gotten formal evidence that this is the smoking gun. We also see that these same fibroblast plugs, as well as tumors, have deficiencies in terms of the immune cells that are infiltrating them. And that includes both macrophage populations, such as F480, as well as lymphocytic populations marked by CD3 or CD4 or CD8. And we see this actually not only in prime fat pads, so these fibroblast plugs, which don't have a tumor, but we also see it in these co-mixing studies, actually, that the amount of lymphocytic as well as macrophage infiltration is significantly reduced. So the immune cells that are infiltrating these tumors, which we believe is actually in this case tumor promoting, but you can also envision may also influence the ability to react to kind of immunotherapies, is actually modulated by the autophagic status of the fibroblasts in these tumor microenvironments. So is this meaningful? Well, we've been actually actively trying to figure this out. And I just have one last piece of data that we did not put in the original G&D paper from last year. And that is actually, we compared these effects. So everything I've shown you has been done in an immunocompetent C57 black 6 mice. This is essentially this co-mixing assay. In this case, it's a specific cohort which we cross-compared the same cell types with a nude mice. And in the context of a nude mice, you don't see these differences. And so, at least in the context of immunodeficiency, these differential effects of autophagic competence versus deficiency on the tumor growth are not observed, suggesting that in addition to these kind of primary roles of what we believe is collagen secretion and the stiffness of the desmoplastic response, there may be an immune component that we do not really understand and are now actively trying to kind of pursue and investigate further. So just to summarize, we believe that autophagy is in for stromal fibroblasts. Bias, the factors that it secretes, is an important and critical aspect of the desmoplastic tumor microenvironment. And we really think this is it kind of floats into two baskets. One is its effects on collagen and ECM proteostasis, which actually influences the ability of these fibroblasts to secrete and stiffen the ECM and then grow tumor cells via its effects on neoangiogenesis, as well as directly on the tumor cells via integrin engagement. At the same time, autophagy is critical with the secretion of a variety of cytokines, including IL-6 that are inflammatory, as well as others that are angiogenic. And then there's other pro-inflammatory cytokines in our profiling that we've seen that we just don't understand that may be actually contributing to these kind of changes in immune cell populations that we're seeing. So I will end there. I want to thank the people who did the work. Jenny Rudnick and Teresa Munkonen really led this study and the in vivo work. At the same time, we've been actively trying to figure out the mechanisms by which autophagy affects secretion, which I didn't really talk about today because we haven't brought it back to the cancer work. We've actually been focusing mostly on aging and neurodegeneration with those studies, but that's been led by Andrew Lytle with help from many others. We've had great collaborators for this study, actually, really, Val Weaver and her expertise of her lab in kind of biophysics of tumor stiffening and mechanotransduction. This is my funding, and this is how you can contact me. And like Christy, even though I'm 15 years out, still actively looking for trainees and postdocs and students. So would love to talk to you if you're interested in any of this work and joining my lab. So I will end there.
Wow, thank you so much. I love these two talks, have been so complimentary and so interesting. I'm going to ask Peter's question first. I'm saving the two Q&A questions to have Christina come back on. But this was a question that I was wondering as well. So you see this amazing effect just by making fibroblasts autophagy deficient. So why can't the tumor cells take on the angiogenic or immune role? So why is it so solely the concern of the fibroblasts in your model?
Yeah, no, I think that's a great question. We do not rule out a role for tumor cells in taking on this role, because the way we did the experiment, we kept the tumor cells intact, right? But if you systemically inhibit autophagy, both the tumor cells as well as the stromal cells will actually doing this. So I should point out our original entry point in this, we showed that inhibiting autophagy in tumor cells affects the cytokine secretion profiles of those tumor cells. So they may be contributory. Why I think fibroblasts may be better at this is because they actually are the master secretors of ECM, which at the end of the day is the infrastructure for vasculogenesis. You can secrete all the IL-6 and VEGF you want. If you don't lay out a collagen network, you're not going to have a vascular genesis. So this is truly a bit of hand waving and frank speculation on my part, but I think it's just that there's more things that fibroblasts secrete that is important for the angiogenic response versus a tumor cell that is mainly secreting factors that endorse that response.
That's a great point. And actually, I'll ask Christina to unmute herself as well, because one of the questions I think that this leads on to is how do you think the intrinsic features of the tumor itself alter? Because obviously polymer middle T is a highly aggressive, fast growing, highly metabolic tumor type. So, how do you think this? How do you envision this in tumors of different types or with different genotypes? Do you think it's as important?
That's a great question. I think that actually, for example, in pancreatic tumor cells, the pancreatic stellate cell has been shown to be important by Alec Kimmelman's lab. It might not, I don't know if they actually looked at the desmoplastic response. They focused on amino acid transfer from the stellate cell to the pancreatic cancer cell. That would be a great place to look, but there may be so many different pathways. The desmoplastic response in pancreatic cancer is just incredibly robust. And you can imagine in such a context that autophagy inhibition may not be enough. You might have to hit many other pathways. The other place where we think about this is actually in non-cancer associated fibrosis. And then actually in those cases, this is a benefit, right? You would want to inhibit autophagy, for example, in liver fibrosis or lung fibrosis. And not our work, but others have shown very similar that you decrease the fibrotic response when you do this experiment in the context of autophagy inhibition only in a specific fibroblast or fibroblast precursor cell, such as a hepatic stellate cell. So I think you're right. It's going to be probably tumor context dependent how important it is. And if I had to speculate, it really is how many pathways the fibroblasts have to kind of build and kind of invoke this desmoplastic response as they grow the tumor. Yeah.
Yeah, I mean, I think Jay answered that question perfectly. I think that's one of the reasons pancreatic cancer is now really starting to move forward as a leading candidate for being targeted with autophagy inhibitors. And I think the desmoplastic response is a large part of that. And I'd argue now, and Jay, I'd love to hear sort of your thoughts too on breast cancer has not really pushed forward as much in clinical trials, but I think a lot of your work suggests perhaps it should. Are you?
Yeah, well, it's a bit complicated because the primary effect, where we actually started with this is actually, could this actually be used as a metastasis prevention strategy? In fact, 10 years ago, everything we did to kind of, quote unquote, test that hypothesis has turned out to be wrong. And it took us, so that was that dev cell paper was like 9 years of us like convincing ourselves, like, why was the original hypothesis so wrong? Like, why is it completely opposite? But I think it actually invokes what you're saying. So one of the experiments we're doing now is that we're actually taking the FSP-CRE model as a metastasis kind of host. And now we're actually putting in tumor cells that are either wild type or autophagy inhibited. So if you remember from the dev cell paper, the autophagy inhibited tumor cells actually grow faster in a normal host, right? They grow and they have metastatic outgrowth at higher rates. So the question is now if you've concomitantly hit the fibroblasts, do you still see that phenotype? And so kind of redoing kind of the work that the fly biologists have. They can mix and match things. Trying to do that in this context as well.
Right, which of course, I mean, you're going to be inhibiting autophagy in all the tissue, systemically, yeah. Right? So.
Yeah.
So, and actually, talking about metastasis, that brings me to one of the other questions is, so would you expect that inhibiting autophagy in fibroblasts would reduce metastasis, specifically like osteoblastic metastasis?
Yeah, so
question since your tumors don't grow. So
yeah. So so we're actually asking that question now, but you're right. So you can't do it through a spontaneous metastasis assay. So you have to do it by a tail vein injection to at least get initial assay, which actually has its own caveats and problems. I actually don't know. I actually really don't know the answer to that because metastatic tumor microenvironments are not as desmoplastic as primary tumor environments. So it's really one of the interesting things about desmoplasia. Desmoplasia occurs like gangbusters in the primary tumor, and that's predictive of what happens in terms of metastatic. But if you actually look at the metastases themselves, they don't really show as much desmoplasia. That it might still be that the fibroblasts need to be activated. And so the question we're asking is that if you don't have an autophagy competent fibroblast, does that even have a genetic role in metastasis? But we may not actually see a kind of traditional desmoplastic response. We're not expecting to for those types of studies. Yeah.
And what about with your experiments, Chrissy? Is that do autophagy defective pancreatic cancer cells or lung cancer cells, are they more prone to metastasis or not so much?
Yeah, so I mean, in our studies, we are just now getting into moving our stuff into more mouse models, and so we can ask those questions. So far, we've really focused on the cell biology, you know, really looking at the mechanisms in vitro, which of course we can all argue is not physiologically relevant, but perhaps the easiest way to get at mechanisms. So we're just now starting to investigate those questions, and I think, you know, again, we're collaborating with Ruben Shaw's lab to build the models, and then we can ask some of those questions a little bit easier.
I mean, I guess as both your talks point out beautifully, it's a really complicated question, right? Because it also is going to come into stroma, aging, tissue specific. Like, for example, is it different with sarcomas? Is it different from those? I mean, it's a very complicated question. But I have to ask this question that came up because every cancer biologist is going to ask this question: what is the P53 status have to do with either of these phenotypes?
Well, I think as in all fields, P53 has become infamous in making things complicated, and there, you know, the autophagy field has not escaped it. And so, you know, there were sort of two papers, seminal papers, that kind of really looked at this a while back now, and that would be Alec Kilman and Kevin Ryan's papers, suggesting originally, I think Kevin Ryan suggested that P53 could affect whether or not autophagy played a role, I guess, in tumorigenesis. And then I think what's been shown, I think pretty nicely, is that it really doesn't. If you think about P53, not like full double, you know, homozygous knockout, which is really not that relevant in tumorigenic, you know, it's really usually a hemizygous loss over time. That it really doesn't seem to play a huge role in whether or not tumors are, I guess, really dependent on autophagy. But again, it's getting a lot more complicated now as we start to look with J stuff and metastasis and things. And so he might be able to speak more to the breast cancer context for P53 mutations.
Yeah, we have not really. So our excuse me, our models are P53 intact. The polymiddle T model, you don't need P53 loss to get a metastatic phenotype. We see, so we've extended some of the pro-metastatic effects of autophagy into cell lines. We have not gone back in vivo. And we see those pro-metastatic differentiation programs in both P53 null or inactivated, as well as P53 competent. So in breast, fortunately, you have cell lines that run the gamut, right? And so we don't see an effect of that pathway, but with regard to like desmoplasia and so on and so forth, yeah, we haven't really even considered it. It's a good point. Yeah.
What about in all those clinical trials where they've been trying out chloroquine or other autophagy inhibitors? Do they see any associations?
No, not so far. Yeah.
That's interesting.
Yeah. I mean, so most of the clinical trials, I think it's important, they have shown promising intermediate biomarkers of various sorts, but they've not really shown an effect on overall survival. And I'll get a drink of water in just a second. I think the big issue there is the pharmacodynamics. So the amount of chloroquine that we can get into a patient, if you actually do the back of the envelope calculations, it's not a very potent effect. Like if you were going to take that concentration of chloroquine and that you see in the tumor microenvironment or in the patient's serum, it's really on the low end of things that we use, both in cell culture as well as in preclinical models. And so I think that what we've learned from the hydroxychloroquine studies is that we're probably not reaching a potent enough autophagy inhibitory effect. And then you throw on top of all of this stuff that, you know, Christina has done, in the sense that you have resistance mechanisms that you know and are expecting, and then, you know, our lab has shown that you may actually increase metastatic events and so on and so forth. So hydroxychloroquine probably, when all is said and done, similar to its infamous role in COVID, it's probably not going to be tenable as a long-term strategy for autophagy inhibition in cancer. And like I said, it speaks to the need for new chemical matter. That's why we need people like Nick to keep making drugs in this area.
Yeah, so I was going to ask that question. I mean, how far are we away then from more potent molecules in the clinic? I mean, as you say, Nick and I'm sure others are working on these things with Ruben and others.
Yes. So I don't know if you want to say anything, Christy. I can give you some color commentary. It slowed a bit because the original trials were not that promising, but I think there's now with the papers that came out about 2 years ago showing the combination of hydroxychloroquine with MEK inhibitors, it reinvigorated some interest. But we're hopeful that inhibitors in other nodes of the pathway, such as ULK1, VPS34, may go forward. Or you get better lysosomal inhibitors that, for some reason, you can actually now not have toxicity but have a greater potency.
Yeah, I was just going to add that Ravi Amravati is designing better lysosome inhibitors that I think could be promising.
But Christy, I mean, you showed in your introduction that there was significant benefit from chloroquine. So are the other inhibitors not recapitulating that, or is there problems with toxicity, or perhaps are there non-autophagy dependent effects of chloroquine?
I think, to Jay's point, I think a large part of it is that we're not really that. Yes, there have been anecdotes that I think are promising, which is why the field still exists. But I think we aren't getting enough drug in. I also think the data suggests that there are groups of patients that really are responding. And so we need better biomarkers to understand what is unique about that group of patients. For maybe it's KRAS mutation. I don't know. I don't think it's P53 mutation. So there's other, perhaps it's not sequencing, right? Maybe it's not the mutational status, but there's other metabolic effects or, unique aspects to the tumor that we need to be able to identify better, because maybe it's a select 10% of patients that are responding, but we don't know which 10% those are, right? So, we're just not there yet. But I think there are anecdotes, and I think anybody that's been on the ground in the clinic treating these patients has anecdotes of patients that have responded really well. We just don't know why.
I mean, this just goes to what a lot of people don't realize, even cancer researchers, is markers of resistance are important, but also markers of response. And I feel like that's often understudied or underfunded. We're almost out of time. Actually, we're past out of time, but I'll let you guys go with the last question, which is sort of kind of space agey. So I don't know how easy it'll be to answer, but sort of putting together all the components of the things that you both have been talking about. Do you envision that in future, maybe many years in future, we would have a patient walking in the clinic and you'd actually take into account their age, their stromal status, how desmoplastic things are before you prescribe a therapeutic regimen to treat their cancer? Or is that just going to be too complex and too difficult to bring into a personalized medicine kind of regime?
Oh, I don't think it will. I think we definitely will be. I mean, 15 years ago, you would have never thought that we'd be taking into a patient's sequencing their tumors into account as to how we treat them. So I think for sure, as we can get more personalized and single cell technology becomes easier, especially for tumors that are accessible, right? Breast tumors and melanomas and things like that, for sure, I think those things will be taken into account. It becomes a little bit harder with tumors that are not accessible, right? Pancreatic cancer and certain types of brain cancer that become a lot harder to access that information. But I would say that for sure, all of the science is leading towards we need to be moving in that direction. And so we need sort of the tools, the devices, the engineering, right, to be able to do that. But I think the field's moving in that way. Do you agree, Jay?
Yeah, I definitely agree. And I think, the autophagy field isn't there yet because what we study is the biology is dictated with the dynamicism of the process, right? It's a dynamic process, and pretty much anything we have as a biomarker, tissue or blood or anything, is a readout of a static kind of snapshot in time. And so that's what we struggle with in the autophagy field. How do you actually, how do you know if you've gotten a great response or a bad response? If, in cells, you need a tandem GFP cherry reporter. You can't do that in a human, right? And so that's one of the struggles. But I agree with Christy in the sense that, we've already seen this, for example, in immunotherapy. So there's a wide bit of granularity in terms of which cells are PD-1 and PD-L1 positive and so on and so forth. I think once we start to identify biomarkers of response that we can start to now multiplex this and with greater single cell and spatial resolution. Even though we are dealing with a dynamic process, we should have enough kind of static surrogates, that we can get that picture that we need to actually utilize this properly.
It's exciting times. All right, I'm gonna let everyone go. Thanks to all the registrants who actually stayed behind for my favorite part of these talks. Just as a reminder, we'll have another one next month, so tune in on that. And then everyone for the trainee roundtable and our two speakers, please just stay online. Though if you need a bio break, that's fine, because we have a couple of minutes, I think, before the trainees sign on. Thank you so much.
Thanks, both of you. I will see you this afternoon. Thanks a lot.